In this video and the next, LTB 3 and 4, we continue the discussion from LTB 2, in which we talked about a very high section of midbrain in the neighbourhood of the diencephalon. The circled area, which is enlarged in the right image, is the image of high midbrain that we saw in LTB 2. It is time for a close look roughly at this axial level. In this video, we will concentrate on pertinent structures along the vertical midline, passing from ventral to dorsal. First, let's talk for a moment about the orientation of the midbrain both in the upper left-hand corner, the MR image, and that of the anatomic image that takes up the majority of the slide. By convention, anatomic images that you might see in a pathology laboratory are oriented such that the ventral portion of a slice of brainstem points towards the bottom of the image and the dorsal portion towards the top. But that's just the reverse of what we see in the radiographic images studied in clinical practice. Easy enough to fix that. Now, as in the MR image, in the anatomical image, ventral midbrain points upward. Now we enlarge the anatomic image. Keep something in mind, however. Not all midbrain axial sections show you the very same internal structures because the level of the axial section differs. The midbrain image to the right is lower than the one to the left. For now, it doesn't matter how these two images differ in specific terms. We will work systematically through various levels. One other thing to mention about all the anatomical images that we use: white matter stains darkly, because myelin takes up the stain avidly. Grey matter and glandular structures are relatively pale. It is helpful to approach images in a systematic way. We will pass from the ventral surface to the dorsal aspect in the vertical midline. Then, in LTB4, we will move from side to side horizontally. Where does the red dot sit? It sits in the interpeduncular fossa, between the two cerebral peduncles on either side of the dot. The cerebral peduncles pass ventrally or upwards, not dorsally as the blue arrows indicate. The red arrow passes through the midbrain heading dorsally, and the arrowhead sits in the cerebral aqueduct, which is otherwise known as the aqueduct of Sylvius. Notice that a white matter tract passes from side to side just dorsal to the cerebral aqueduct, as marked by the bidirectional yellow arrow. This posterior commissure contains fibres that connect pretectal areas on either side of the midline. The term pretectal refers to an area that we visualize superior or rostral to the main structures of the midbrain tectum, or roof. And by midbrain tectum, we mainly refer to what is called the quadrigeminal plate, about which more in LTB5. The significance of the posterior commissure is straightforward. It is involved in the consensual pupillary light response, in which light delivered to one pupil constricts both pupils. Surrounding the cerebral aqueduct is grey matter as indicated by the red arrows, but we should mention a few very specific things about grey matter or nuclei at this level of midbrain. Inside the rough boundary of the yellow box, there are nuclei within the central grey matter and nuclei outside of it. We will mention two pairs of nuclei by name. First, the nuclei of dark Scheiwitz within the central grey matter can be found on either side of the cerebral aqueduct. We would need a cell stain at a higher magnification to see the actual nuclei. Second, the interstitial nuclei of Cajal, just lateral to the central grey on either side, more or less buried in white matter. The white matter contains fibres of the rostral interstitial medial longitudinal fasciculus, or RIMLF. Both pairs of nuclei and the RIMLF are all involved in control of conjugate eye movements in the vertical plane. Where does the red dot sit now outside the dorsal border of the midbrain? It sits in the pineal gland, which is an epithalamic structure. As said in LTB2, given the anatomic proximity of the high midbrain and diencephalon, it should not surprise that we visualize structures of diencephalon along with midbrain at this high level. This last slide of LTB3 is for extra credit, and you could skip it, although I hope you won't. There are structures that we did not identify during our tour of the vertical midline, but they might strike the eye. Looking at the three circled areas from dorsal to ventral, we have the pineal gland in red, as we identified in the previous slide. Notice some very dark specks of calcium in the gland. It is a common finding in the pineal, and you recall that it is an epithalamic structure. Inside the yellow oval within the central grey, ventral to the cerebral aqueduct, is actually part of hypothalamus, one of the four strata of diencephalon that we mentioned in LTB too. So what is the thing in the midline circled in blue? It is not likely an artifact, although our images often have artifacts. It seems like a nuclear structure surrounded by some white matter. It sits in the midline. As it happens, there is a fiber connection between an epithalamic structure that is not the pineal gland, called the habenula, and the midline of midbrain. Bilateral fiber tracts from habenula, each called either the fasciculus retroflexus or the habenular interpeduncular tract, converge at the most ventral portion of midbrain between the cerebral peduncles at the so-called interpeduncular nucleus. We see fasciculus retroflexus and perhaps a bit of interpeduncular nucleus in the blue circle. Like the pineal gland, habenula derives from epithalamus. It receives input from limbic structures. Its neurons activate under adverse conditions, and it may influence other midline brainstem structures. The LTB series is meant to appeal to as many levels of learners as possible. Yes, we are getting detailed. But, as we will continue to see in our next video, LTB4, which takes us horizontally across the high midbrain, there is a systematic way to learn the brainstem really, really well. As always, thanks for watching.